Mic test, sound check, sound check. Hey there, guys. Welcome. This is Alan Joshua. You can call me Sir Joshua, and welcome to another lecture topic of the molecular biology in diagnostics. For this video, we're going to talk about the amplification of nucleic acids. This PowerPoint presentation is prepared by Miss Kathleen May Cruz. Presented by yours truly. Tara, samahan niyo ko. For the outline, here are the subtopics that we're going to talk about. Number 1, the conventional amplification technique, the polymerase chain reaction, modified nucleic acid amplification techniques, parameters that affect your PCR, quantitative PCR, DNA intercalating dyes for qPCR, your fluorophore-labeled oligonucleotides for qPCR, fluorophore-labeled oligonucleotides nucleic acid analogs, and of course, we have the isothermal amplification techniques. Tara, let's start. I have here, ladies and gentlemen, an animated photo of our thermal cycler. This is one of the main equipment that we use for your PCR, or the polymerase chain reaction. When you say polymerase chain reaction, ladies and gentlemen, this is sometimes called as your molecular photocopying. PCR, or the polymerase chain reaction, is a fast and inexpensive technique used to amplify. And when I say amplify, it means to copy small segments or specific regions of your DNA. This is a new molecular technique that involves in vitro enzymatic replication of the defined DNA sequences. Dito. Please do remember, ladies and gentlemen, that when we say in vitro, it happens in a man-made setting, correct? Going back to what we've talked about in one of our previous discussions, when you say in vivo, it's a Latin term that has a literal translation of within the living or within the living organism. But when you say in vitro, it is a Latin term na ang ibig sabihin is in glass. Tama po ba? Yes po. Okay? Ma'am, sir, dito. Your PCR is often heralded or dubbed as one of the most important scientific advances in the field of molecular biology. And take note, today, PCR is used to diagnose diseases, identify organisms such as your bacteria and viruses, match criminals to crime scenes, and it is widely used in biotechnology. Sa madaling salita, when we say PCR, this pertains to a technique to make millions of copies of specific DNA region in vitro. It relies on a thermostable DNA polymerase that requires DNA primers, designed specifically for the DNA region of interest. And this reaction is repeatedly cycled through a series of temperature changes, allowing many copies of the target region to be produced. That, ladies and gentlemen, is PCR. And that concludes our presentation. Salamat po, paalam. Joke lang. And now, for the million-dollar question, how? How does PCR work? Well, in a typical PCR experiment, the target DNA is mixed with 1. Your Taq polymerase. And I think you guys are familiar with your Taq polymerase, correct? Taq polymerase is an example of a DNA polymerase. And this particular polymerase is harvested from a bacteria called your Thermus aquaticus. Thermus aquaticus is a thermophilic organism na makikita natin sa mga hot spring. Totoo, Sir Joshua. Yes, Taq polymerase is a DNA polymerase. Hindi pwedeng kalimutan, kailangan tandaan, ilagay sa puso at isipan. Dito, aside from your DNA polymerase in the form of your Taq polymerase, you also have 2 oligonucleotide primers, and of course, a supply of your dNTPs, all of which, ladies and gentlemen, are found or mixed in a buffer. Hingi ako ng favor. Sir Joshua, question, what are the 3 PCR reaction steps? Hindi pwedeng kalimutan ito. Isaksak mo ulit sa puso at isipan. Highlight, bilugan sa lungguhitan hanggang sa mabutas-butas ang papel mo. You have your denaturation, which is the first step. you have your annealing, and you have your extension. Again, denaturation, annealing, and extension. Paulit-ulit. Again, denaturation, annealing, and extension. Hindi pwedeng kalimutan. Bakit? Uy, basta dadaanan natin yan. Guys, no, dito ano meron. So, joke lang po yan. Guys, no, dito kailangan tandaan mo yan. Hindi pwedeng kalimutan yan. Ako masamang panaginip mo, uulit-ulitin ko yan sa'yo mamaya. Okay? Maski habang natutulog ka na, sasabihin mo, denaturation, annealing, extension, kailangan tandaan. Dito tayo. Sir Joshua, sino-sino nga ulit yung steps na kailangan namin tandaan for the polymerase chain reaction? Well, number 1, you have your letter D, 2, A, and 3, your E. D-A-E. Denaturation, annealing, and extension. For the denaturation to occur, ladies and gentlemen, kailangan ang temperature natin mataas, approximately 94 to 96 degrees Celsius para maghiwalay yung double-stranded DNA natin. Totoo? Yes, Sir Joshua question, saan galing yung double-stranded DNA? Template na tinatawag. Well, your template came from the samples that you have extracted. Tama po ba yun? Okay. Yun yung galing sa patient sample. Yan yung galing sa cell na nilyse natin. Tama po ba? Yes po. Okay. Number 2, you have your annealing. And your oligonucleotide primers play a huge role in this step of the PCR. But take note na ang temperature, ladies and gentlemen, should be lower to 50 to 60 or 50 to 65 degrees Celsius para po kumapit yung mga primers natin sa ating mga complementary sequences. Sir Joshua, after the annealing process, you have your polymerase extension. Sino may pakana ng extension? It's your polymerase. It's your Taq polymerase. Tama po ba? And in order for your Taq polymerase to work, kailangan ang temperature should be raised into 72 to 74 degrees Celsius. Totoo, Sir Joshua? Yes po. Dito tayo. Paulit-ulit ako. Akong masamang panaginip mo. You have your denaturation, annealing, and extension. Going back, when you say denaturation, kailangan ng temperature mataas, approximately 94 degrees Celsius or 94 to 96 degrees Celsius for 1 to several minutes para mag-separate, maghiwalay ang double-stranded DNA mo. Maging single-stranded na sila. Tama po ba? Yes po. Now, for the annealing, ladies and gentlemen, remember your oligonucleotide primers. In order for them to take an effect, kailangan bumaba ang temperature from 50 to 60 or 50 to 65 degrees Celsius. Uy, kumapit yung mga primers dun sa single-stranded DNA natin. What will happen next? Papasok ngayon sa eksena yung ating polymerase, your Taq polymerase, which is again, paulit-ulit ako masamang panaginip mo. Sabi natin, your Taq polymerase is harvested from their thermophilic bacteria called Thermus aquaticus. Tama? Uy, gagana. Gagana ang ating Thermus aquaticus polymerase o ang Taq polymerase kapag ang temperature is at 72 to 74 degrees Celsius. Siya yung magfa-facilitate ng elongation or extension ng ating single-strand DNA. Tama po ba? Yes po. Nung tinatawag nating template, yung in-extract natin kanina. Well, not really kanina. Yung in-extract natin. Okay? Take note, ate, kuya, nanay, tatay. Again, it starts with the denaturation, followed by annealing, and then extension, and then the cycle goes on. Okay? Okay, so I have here an example of a protocol in which we utilize for the thermal cycler in order for us to amplify a certain DNA region. So take note that this will vary depending on the kit or the reagent that have been used. And number 2, it will vary depending on the brand of the machine of the equipment that is being used. Okay, so as you can see here, we have the initialization step. Some machines call it as the initial denaturation step, which makes sense because it happens at 95 degrees Celsius. For this protocol, the initialization or the initial denaturation happens at 95 degrees Celsius for about 2 minutes. There are these machines, there are these equipment that last for a minute lang. Okay, and then for the next step, we have, of course, the denaturation process. Sorry, for the denaturation, sorry talaga. For the denaturation, ladies and gentlemen, it happens for about 15 seconds. And of course, the next step, we have the annealing, wherein it happens at 60 degrees Celsius for 15 seconds. You have the extension, 72 degrees Celsius, and lasts for about 30 seconds. Take note, ladies and gentlemen, that this three steps, denaturation, annealing, and extension, happens for about 35 cycles. Okay? 35 na beses na uulit-ulitin itong tatlong step na ito. Okay? So as you can see here, the initialization happens for one cycle. It happens. Then you have your denaturation, your annealing, and extension, uulit na naman siya. Okay? Ilang beses ang ulit? 35 times. Kapag natungtong na yung pang 35 na ulit, pang 35 na denaturation, pang 35 na annealing, pang 35 na extension, tsaka siya pupunta dito sa next step. Okay? And that next step, ladies and gentlemen, is what you call the final extension. Okay, so that will depend on the type of the machine and the type of reagent that is being used. For this example that I have used, merong pang-apat na step, that is for the fifth step, you have the final extension, which happens at 72 degrees Celsius for 1 minute. Okay, and then you have here the hold. Okay, dito po nangyayari yung data collection. Actually, sometimes there are these machines wherein the data is being collected at this fifth step. Okay, but this protocol, ladies and gentlemen, utilizes a sixth step, which is the hold. Sometimes it's not 10 degrees Celsius. Most of the time it's 4 degrees Celsius. Okay. Okay, so let's say, for example, I have here an ongoing PCR run, and we're going to make use of the protocol that I have mentioned in the previous slide. So we are having the initialization, which happens at 95 degrees Celsius, that lasts for 2 minutes. And then we go with the first step of the polymerization. You have the denaturation. So you have here your template strands. Okay, double-stranded DNA yan. And please do take note, ladies and gentlemen, that if we are talking about in vitro study, if we are talking about your PCR, we call this strand as your template strands. So we don't call them as the parent strands, because I think when you say parent strands, it's more appropriate to use the parent strand term in addressing the replication that happens inside the nucleus, an in vivo replication. Okay? So when you say template strand, okay, sa labas nangyayari yan. But when you say parent strand, yun nangyayari saan? Doon sa loob ng nucleus mo, which happens within your cell. Okay? Okay, sana nakuha yun. No, dito tayo. Wala lang, kaartihan ko lang. So again, denaturation happens at what temperature, Sir Joshua? It occurs at 95 degrees Celsius, wherein this temperature facilitates the breakage of the hydrogen bonding between your nucleotide base pairs. Tama po ba? Remember, you have your ATGC pairings. Masisira yung hydrogen bond between them, kaya sila maghihiwalay, creating a ssDNA, creating your single-stranded DNA templates. Okay? Now, for the next one, kailangan bumaba ng temperature to 60 degrees Celsius in order for us to go to the annealing step. When you say annealing, ladies and gentlemen, we need your primers, your oligonucleotide primers. And in order for these primers to bind, ladies and gentlemen, kailangan ng temperature is at 60 degrees Celsius. Tama po ba? Yes po. Primers will bind to our template strand. And then we're going to proceed with the next step, which is the extension. For the extension process, napaka-importante ng role ng ating Taq polymerase dito. And in order for a Taq polymerase to produce a reaction, kailangan ang temperature is within or at 72 degrees Celsius. This green crescent represents our Taq polymerase, which is of course harvested from your Thermus aquaticus organism. Siya po magfa-facilitate ng extension natin. And now you have here a double-stranded DNA. Okay? So, yung double-stranded DNA na yan, ladies and gentlemen, magsisilbing template ulit yan. Okay? Para po ma-facilitate yung pagdami. Para makagawa tayo ng millions of copies of this DNA region. So, it will undergo again denaturation, annealing, and extension. And the cycle repeats over and over again. Okay, so denaturation, annealing, extension, paulit-ulit na mangyayari yan hanggang sa ma-reach mo yung final na cycle. Okay. Okay, so as you can see here in the slide, yung thermal cycler daw po natin is usually set to run for 30 cycles. Okay? But in my example, I think I was able to give you guys 35 cycles. Tama po ba? Yes po. So dito, after 30 cycles, according to our reference textbook, there will be more than 1 billion short products derived from each starting molecule. And again, we call these products as your amplicons. And these amplicons are analyzed pwedeng via gel electrophoresis or pwedeng diretso na doon sa software na meron yung laboratory nyo. Okay? Okay, so now we have here the necessary requirements for polymerase chain reaction. I don't know if my English is correct, no? Necessary requirements. I don't know if that's redundant, but bahala kayo sa buhay nyo. Basta, these are the requirements in order for us to produce the polymerizing chain reaction or the polymerase chain reaction. Okay? So, number one, kailangan natin itong equipment na to, and we call this your thermal cycler or your thermocycler or your PCR machine. This equipment provides the necessary temperature changes in order for us to achieve the denaturation, annealing, and extension. Tama po ba? Yes. And then we have, of course, your DNA template or your target DNA or your DNA source. This will be coming from your blood, your stool sample, your urine sample, your saliva, your hair, kahit ano, basta yung pinanggalingan ng specimen po natin. Diba ito po yung in-extractan natin, kinuhanan natin ng nucleic acid. Diba, naalaling 4 na steps in order for us to isolate the template or the eluate na tinatawag. You have your cell lysis, the adsorption or the binding, then you have your washing, and then the last step is the elution step. Okay, especially for making use of our spin columns. And then we have your primers. Ang primers po siya yung nagdidictate ng specificity ng reaction natin. So you have 2 primers. You have the forward and you have the reverse primers. Then of course you have your DNA polymerase. Okay, sino nga ulit yung DNA polymerase na ginagamit natin? It's your Taq polymerase. Taq polymerase is a really thermostable type of polymerase harvested from your organism called Thermus aquaticus. Tama po ba? Yes, and we have your DNTPs. Your DNTPs contains the nucleosides and your triphosphate groups. Basically, these are the building blocks from which your Taq polymerase will synthesize a new DNA strand. Okay, and then you have your buffer, of course, your monovalent or bivalent cations. You have your nucleotides, you have your PCR water. And take note that if we are talking about your PCR water, this is the same with your nuclease-free water. And when you say nuclease, di ba these are enzymes that degrades your nucleic acids. You have your DNases and you have your RNases. These enzymes are important. Importanteng wala sila doon sa iyong water because they degrade your nucleic acids, and you don't want that because it destroys your starting material. It destroys your target DNA or destroys your DNA templates. Okay? Ayun. Okay, so I have here, ladies and gentlemen, photos of your thermal cyclers. Sabi ko sa inyo, yung thermal cycler natin is the main equipment that is used in order for us to produce PCR products. Tama po ba? Yes po. Ngayon, you have here conventional PCR machine. You have your real-time PCR machine. Both of these are thermal cyclers, aka thermocyclers, and these are from Biorad Laboratories. Kapag binuksan mo yung dalawang yan, ganito yung makikita mo. May makikita ka mga butas-butas dito. Kasi dyan po natin ilalagay yung ating 96 well plate na tinatawag. Okay? O kaya yung strip mo kung anong gamit ninyo. When I say 96 well plate or PCR strip, doon po natin nilalagay yung reagent together with the template. Okay? Kasi doon nangyayari yung reaction. Okay? So, anong meron dito? Anong pinagkaiba, Sir Joshua, ng conventional PCR machine sa real-time PCR machine? Well, both of these, ladies and gentlemen, is capable of providing the required temperature for the reaction. Remember, nag-vary in temperature. You have your initialization, denaturation, annealing extension. Then you have your final extension pa where the data is being collected by the real-time PCR machine. Okay? But thing is, ladies and gentlemen, dito, ang pinagkaiba nila, okay, kaya po nila mag-provide ng certain temperatures for the different steps of PCR. But yung conventional PCR machine po natin, hindi niya kaya mag-analyze ng product. Okay? Bale, ang gagawin mo lang dito, you have your reagent, halo mo sa iyong template, then you're going to place it in your tube or in your wells or in your strip, then lagay mo dito for a couple of hours, matatapos yung cycles, tsaka mo lang siya pwede i-analyze. Usually via agarose gel electrophoresis. Okay? But for your real-time PCR, maski ongoing ang running, hindi pa natatapos yung running, hindi pa natatapos yung cycle, pwedeng makita mo na kung sino yung merong ganitong DNA sa sample. Kaya pala siya tinawag na real-time. Kasi makikita mo agad, maski hindi pa tapos yung running, kung sino yung nagpa-positive, kung sino yung nagne-negative. Totoo, Sir Joshua? Yes. Okay. Real-time PCR, kaya niyang i-analyze yung PCR products mo. But for your conventional PCR, you still have to analyze it using a separate test. Okay? And that test is usually the agarose gel electrophoresis. Okay? So, ayun, real-time and conventional PCR. Siyempre, mas okay itong real-time kasi all-in-one na yan. Siya na bahala magsabi kung sino merong ganitong DNA, sinong ganitong wala. Okay? Conventional, you still have to analyze the PCR products. Wala lang, na-share ko lang. Ayun. Okay, and we have here the common components of the Pcr Master Mix. As you can see here, ladies and gentlemen, you have your component, you have here the volume, and you have here the final concentration of each component used in the master mix. But take note that if ever in the future you decided to work in a molecular laboratory, mapapansin mo, hindi naman porket ito yung nakasulat dito sa slide na to, ay yun yung gagawin nyo doon sa pagtatrabaho nyo. Because the concentration, the volume of a component of the master mix will depend on the reagent. Okay? Merong manufacturer's guide yan. Okay? May manufacturer's manual. In order for you to know the needed or the required volume or the required concentration per component of the master mix. Okay? So again, this is just an example. Okay? This will vary depending on the brand of your kit or your reagent. Okay? Okay, so I have here, ladies and gentlemen, the standard 3-step Pcr cycling. Ha? Sir Joshua, sabi mo sa amin, nagpakita ka kanina ng protocol, 6 yung steps na yun. Well, relax, relax, calm down, calm down. Anong meron? Same naman yon. Kanina nabanggit natin na ilang steps yon, 6, which makes sense because in the 3-step Pcr cycling, the first step is still the initialization or the initial denaturation, followed by the 3 steps in PCR, the denaturation, annealing, and extension. Uy, sila yung second step dito sa standard 3-step PCR cycling natin, which makes sense because itong tatlong step na to, itong DAE natin is repeated for many times. Okay, uulit-ulitin siya. Nakalagay din sa example natin kanina, it's 35 cycles, correct? Yes, but again, dito, 25 to 35, ano ba talaga totoo? Relax ulit, calm down, calm down. Anong meron? Sabi natin, ladies and gentlemen, the number of cycles employed in PCR cycling will depend on the kit that is being used. Okay? Actually, meron pang 40 cycles yan. Iba tumatagal ng 42 cycles. Again, that will vary depending on the reagent's instruction. Okay? Ayun. And then you have here the final extension, diba? And I think a while ago, yung final extension natin occurs for about 1 minute. Dun sa example na binigay ko sa inyo, ilang cycle? Isang cycle ulit. And then you have the hold. And I think I was able to give an example of 10 degrees Celsius. Tama po ba yun? Yes po. Okay. Again, magdedepende yung temperature and magdedepende yung number of cycles doon sa reagent. Kasi meron na nakalagay na instructions doon. But if not, kinakalculate po yung temperature na yan. Basta tatandaan nyo, kaya pala Sir Josh, may mga ranges na nakalagay. May nakalagay 94 to 96, mga ganon. Okay? So, kinakalculate po yan. Okay? So, again, these are the standard 3-step PCR cycling. Initialization or initial denaturation. You have your DAE na inuulit over ilang cycles. Then you have the final extension and the hold. Okay, so I have here a video which is lifted from one of the laboratory videos that I made for you guys. So it's going to show you guys, kasi kanina pa ako kuda ng kuda. Ano gagawin mo? Paghahaluin mo yung master mix, tapos yung template mo, tapos lalagyan sa PCR. This is what happens. Okay, so this is an animation, an animation of the different steps before loading it into our thermal cycler. So as you can see, meron ka ditong master mix in yung reagent mo. Halo-halo na yan. You're going to place it into this 96 well plate. 96 kasi 96 yung wells niya. 96 yung butas na to na kung saan lalagyan mo ng reagent. And syempre, hahaluan po natin yan ng DNA template. And yung DNA template po natin, syempre, that came from your sample. Okay, so isang DNA template, patient ni ganito sa well A1. Patient ni ganito naman sa well A2, and so on and so forth. Okay? So for the next step, you're going to place it in your thermal cycler. Again, when you say thermal cycler, this is also known as your PCR machine or your thermocycler. Pag binuksan mo yan, ganito yung itsura, papasok mo lang dito. Okay? Ayan, sakto for 96 wells. And then, siya na bahala, ladies and gentlemen, mag-facilitate ng temperatures na kailangan. Okay? Remember your initialization, denaturation, annealing, extension, final extension. Siya na bahala mag-provide ng temperatures for that. Okay? Ayan. And then after 1 cycle, magkakaroon ng yung DAE natin. So yung kung merong isang pirasong DNA strand dyan, mga dodoble sila. Okay, remember exponential, yung pagdami ng mga DNA strands dyan sa iyong sample. Okay? Okay. So, anong gagawin susunod dyan, Sir Joshua? Well, tapos na tayo doon sa thermal cycler. Anong gagawin natin? Well, basically, we're going to analyze the product. Lalo na kung conventional, katulad nito. Kung conventional yung PCR machine natin, we're going to analyze the product usually with your agarose gel electrophoresis. Okay? Okay, so now we analyze your PCR product. Okay po? So sabi natin, kung conventional PCR machine ang gamit natin, we analyze the product usually with the use of your agarose gel electrophoresis. Okay? Kung real time, there's no need na. Nagiging optional na lang. Kung gusto mo pang i-subject yung samples na lagay mo sa real-time PCR machine sa agarose gel electrophoresis. Okay? So, again, kung conventional ang gamit natin PCR machine, then we analyze the product using your agarose gel electrophoresis. Okay? Bakit yan, Sir Joshua? Well, yun kasi yung pinaka-common. It's the simplest and the easiest procedure daw in order for us to check for the PCR products. Okay? Ang pangit lang dito is that we sometimes use intercalating dyes that are carcinogenic or toxic. Okay? Yan lang. Tapos medyo hindi ganun kataas ang throughput nito. Okay? Again, when you say throughput in your clinical chemistry, na daanan nito o kung saan man, isa sa mga lectures natin. Diba, when you say throughput, ito yung dami ng samples na kaya mong i-process within a given period of time. Okay? So, again, this is your agarose gel, as you can see, and dito sa extreme left ng agarose gel natin, you have these rectangular holes. Sometimes this is square. Okay? Basta dyan natin ilalagay yung sample na sinubject natin sa thermal cycler. And usually our sample is incorporated with an ethidium bromide intercalating stain. Common na ginagamit na stain for this purpose is your ethidium bromide. Sometimes we make use of the gel red, you have your cyber green, etc. etc. Okay? So, anong meron pagkalagay natin dito? Well, kailangan mag-migrate yung mga molecules ng nucleic acid mo na nandito to your right. Okay? Kailangan magmamigrate sila from here to here. Paano mangyayari yun? Number one, porous kasi yung gel natin. It's porous, meaning marami siyang microscopic holes. Okay? Okay. May butas-butas pala to. Yes, that will facilitate the migration of your DNA molecules. Now, Sir Joshua, magmamigrate ba siya ng kusa? No po. Kaya kailangan natin dito ng electricity. Kaya natin siya ilalagay sa electrophoretic chamber. Magbibigay tayo ng electricity, magfo-flow dito sa agarose gel natin. So, itong mga DNA molecules, mga nucleic acid molecules natin, in the presence of electricity, they will migrate towards the positive electrode. Okay? So, ang principle kasi ng agarose gel electrophoresis is that yung bilis ng migration is proportional to the size of the molecules. Meaning, the smaller the molecules size are, then mas mabilis silang gumapang dito sa gel natin. Okay? Para saan yung dye? Para saan yung intercalating dye? Para makita natin, okay, now it makes sense. Okay po? Tandaan natin, ladies and gentlemen, that if the expected band is present, ibig sabihin, successful yung PCR reaction na ginawa. Okay? Kung hindi po makita yung band dyan, baka wala kang na-amplify. May nakalimutan tayo ilagay sa reagent, hindi ka nakapaglagay ng template, or hindi maganda yung extraction na ginawa mo. Okay? It's always a case-to-case basis. Okay? Merong troubleshooting for that. Okay po? Okay, and now we have the different variations of the polymerase chain reaction. Maraming modifications ang PCR. Okay, you have your reverse transcriptase PCR, you have your nested PCR, you have your multiplex PCR, and you have your digital polymerase chain reaction. Okay, so all of which, ladies and gentlemen, can be done with the use of your conventional PCR, and all of which can also be done with the use of your real-time PCR machine. Okay, so again, ang pinagkaiba ng conventional PCR machine sa real-time PCR machine mo is that your conventional PCR machine kailangan mo pang i-analyze yung PCR products manually. But for real-time, no need na. Okay, it's no longer necessary for you to analyze the PCR products with the use of your agarose gel electrophoresis because the interpretation, the analysis is conducted by the machine. Okay, so dito tayo. Okay, so let's start with your reverse transcriptase polymerase chain reaction. Familiar? Naku, alam ko na pag-usapan natin to in one of your video discussion in your laboratory, correct? Yes po. You have your reverse transcriptase polymerase chain reaction. Sabi natin, ladies and gentlemen, itong modification or itong variation ng PCR na to ang ginagamit natin if we want to amplify, if we want to make millions of copies of your RNA targets. Totoo, Sir Joshua? Yes. So are you telling us that this particular variant of PCR is used if we want to check for RNA viruses in our samples? Yes po. Oh, so are you telling us that this particular PCR method ang ginagamit natin for SARS-CoV-2 testing? When you say SARS-CoV-2, yan po yung causative agent ng COVID-19 natin. Like, oh my gulay. Like, oh my gulay talaga. Yes po. Okay? Again, ito yung ginagamit na PCR method or PCR variant or PCR modification to check for RNA targets. Okay? But, ano po bang pinagkaiba nito, Sir Joshua, mula sa conventional o dun sa regular na PCR? Well, di ba kapag PCR, pinag-uusapan natin, tatlong major steps yan. Yes po, you have your denaturation, annealing, and extension. Paulit-ulit yan. Ngayon, for reverse transcriptase polymerase chain reaction, may dinagdag tayong step. Ano yun? Alam mo to, napag-usapan natin to ay Sir Joshua nga, we remember now. It brings back so many memories. Yes, everybody hurts sometimes. Everybody hurts someday. A. But everything's going to be alright. Go and raise a glass and say, A. Here's to the... Hindi, joke lang po. Sorry, pasensya na. Nakikarried away na naman ako. Guys, no, dito. Ano meron? Shocks class, no shocks. Sabi natin, ladies and gentlemen, I think we were able to talk about this in your central dogma. Diba? You have your DNA which undergoes replication. Tama? And if you want to produce proteins from that DNA, you need to first transcribe your DNA which is found in your nucleus into an mRNA. Tama? What do you call that process nga ulit? You call it transcription. Tataa? Yes. Question. If we want to produce DNA from RNA, what do you call that step? It's called reverse transcription. Tama po ba? Yes po. Reverse transcription. And that reverse transcription step, ladies and gentlemen, is possible with the help of an enzyme that is harvested from your RNA viruses. Sino yan? Yung reverse transcriptase enzyme natin. Sabi ko sa inyo, paulit-ulit ako masamang panaginip mo kapag sinabing mga ACE, mga enzyme yan. Okay? Totoo? Yes. Oh my gulay. Basta, tandaan mo yung ACE hardware, hindi enzyme yon. Gel ACE, hindi rin enzyme yon. Basta yung ibang ACE dyan. Promise. Guys, no, bahala kayo sa buhay ninyo. So again, ladies and gentlemen, kailangan muna natin i-convert yung ating RNA into becoming a DNA bago po ma-proceed yung amplification. Kasi kung RNA lang yan, walang mangyayari dyan. Kailangan ang RNA mo, ladies and gentlemen, na ma-convert into anong cDNA, Sir Joshua? Uy, nabanggit ko na to sa inyo. Tama? Let's go back. Let's go back to what we've talked about. Ano nga ibig sabihin ng cDNA? Let's go back. Let's go back to the Old Town Road. I'm gonna ride till I can't no more. I'm gonna take my horse to the Old Town Road. I'm gonna ride. Joke lang po talaga yan. I'm sorry. Nakikarried away na naman ako. Guys, so dito, again, when you say cDNA, it stands for complementary DNA. Okay, so you have your RNA targets. Kailangan mag-undergo ng reverse transcription. Sino may pakana nito? Yung enzyme na galing sa RNA viruses natin, you call it reverse transcriptase. Siya may pakana ng conversion ng RNA into your complementary DNA or the cDNA. At yung cDNA na yan, ladies and gentlemen, siya yung magsisilbing template para dumami na dumami yan. It will undergo denaturation, annealing, extension, and it's going to produce like millions of copies, millions or billions of your cDNA. Okay? Tandaan mo to, huwag kakalimutan, kailangan tandaan, isaksak sa puso't isipan, reverse transcriptase, highlight, bilugan sa lungguhitan hanggang sa mabutas-butas ang papel. Dito tayo. So I have here, ladies and gentlemen, a photo which I got from our reference textbook. So you have here your RNA. And then, in order for you to amplify these RNA targets, ladies and gentlemen, kailangan mag-undergo muna siya ng process called reverse transcription with the help of what enzyme again? Reverse transcriptase. Your RNA will be converted into your cDNA or your complementary DNA. And your complementary DNA, ladies and gentlemen, will then undergo your denaturation, annealing, extension. Tama po ba? With the help of repeated cycles, dadami at dadami yan, you will produce millions or even billions of copies of your cDNA. Okay? Okay, now for the next one, ladies and gentlemen, we have the nested polymerase chain reaction. This is another modification of your nucleic acid amplification technique, correct? And for this nested PCR, I want you guys to remember three things. Number one, gumagamit po tayo dito ng dalawang set ng primers, okay? So yung first set ng primers natin has the forward and reverse. Okay, and the second set of primers also have the forward and reverse primers. Okay, and Sir Joshua, you told us in one of the previous slides that primers dictate the specificity of the reaction. Totoo po ba yun? Yes. So are you telling us, Sir Joshua, that this nested PCR is more specific compared to the regular PCR we do? Oh yes. Like, oh my gulay. Okay, so mas specific ito because of the two sets of primers. Number two, the master mix template mixture is subjected into 2 rounds of PCR. Sir Joshua, 2 beses sinasubject sa thermal cycler yung ating sample at yung master mix. Yes po. Okay, so 2 beses tayo nagpi-PCR, 2 beses tayo nag-thermal cycler dito. Yes po. Okay, so with this, ladies and gentlemen, with this 2 times or 2 rounds of thermal cycling or of amplification, this only increases the sensitivity of the PCR, the sensitivity of the reaction. Yes, so are you telling us that this is more sensitive compared to other PCRs? Yes, this is more specific because of the 2 sets of primers, and this is more sensitive because of the 2 rounds of amplification. Ang pangit lang for your nested PCR is that this is prone to contamination. Imagine, nakasil yung plato mo, tapos ininit-init mo. Possible po, ladies and gentlemen, na pag binuksan mo yung plate mo later on, after the first amplification, after the first round of amplification, kumalat sa air yung tinatawag nating amplicons. Are you guys familiar with amplicons? Of course, intercourse, you know amplicons are. You know what amplicons are. Amplicons are the amplified product after thermal cycling, like, oh my God, kumakapit sa damit, sa buhok, sa kung ano-anong parte ng katawan mo at sumasama sa hangin. Ay, okay. So this may cause contamination. Yes. Tama. Yun yung problema kay nested PCR. Okay, it may contaminate the second tube kasi merong step dito. Kasi bakit mo babuksan yun? Pwede bang hindi nilang buksan? Hindi, kailangan mo talaga siyang buksan. Kasi nga sabi natin, gumagamit tayo ng 2 rounds ng primers. 2 rounds ng primers. 2 sets ng primers and 2 rounds of amplification. So, ang may step kasi dito, may part kasi dito na kukunin mo yung first round product, tapos ilalagay natin sa pangalawang tube that contains your second set of primers. Okay. But sabi nila, ladies and gentlemen, according to our reference textbook, this may be avoided by physically separating the first and the second round amplification mixtures by using wax or oil. Okay? Or, pwedeng gawin yung isang step sa ibang room and yung separation, pwedeng yung separation gawin sa isang room and yung paglalagay naman doon sa pangalawang tube gawin sa isa pang room. Okay, so medyo hassle lang ng konti kasi you need 2 separate rooms for this. And, super prone sa contamination. Ayawan na kapag nagkaroon ng contamination sa molecular biology laboratory. As in, ayawan na. Kasi kapag nagkaroon ng contamination dyan, iis-isin nyo lahat ng sulok, lahat ng gilid ng room with bleach. Promise, ayaw mo nun kasi iyak-iyak ka na, ang sakit sa lalamunan, ang sakit sa ilong, masinghot nung amoy ng bleach. Maski 10% lang yan, ayaw mo nun. Nakakaiyak yun. Promise, I'm not even joking, I'm not exaggerating, maiiyak ka. I have experienced that kasi nagkaroon ng contamination. Wala lang, na-share ko lang sa inyo, okay? So that's for your that's for your nested polymerase chain reaction, okay? Thank you, Nest. Thank you, Nest. Bahala kayo sa buhay niyo. Okay, thank you. Nest, dito. I have your photo which I got from our reference textbook. You have your traditional nested PCR and you have your semi-nested asymmetrical PCR. Like I said, ladies and gentlemen, in your traditional nested PCR, you make use of 2 sets of your primers and dalawang beses natin sinasubject sa thermal cycling. Ang pangit lang, it may cause or it is prone to contamination. Okay, so as you can see here, ladies and gentlemen, you have here your template. Tama po ba? So anong meron? Gumagamit tayo ng dalawang set ng primers. Yung unang set ng primers is what you call your outer primers. This is also known as your outer primers. But when you say second set ng primers, this is also known as your inner primers. Okay, so you have here your template DNA. You have here your amplicon. This is your first round product, or this is your first amplicon. And this first amplicon is needed for the second round of amplification. Okay? So as you can see, it contains the DNA region of interest or the sequence of interest. Itong part na to. So, sasubject natin tong first round ng amplicon sa panibagong tube that contains the second inner primers or the second set of primers. Okay? So, since, ladies and gentlemen, you have a second set of primers, this is your template. So, ang gagawin, maghihiwala ito due to denaturation, tapos mag-anneal and all that stuff. Yung primers natin, yung second primers or yung inner primers natin will only make a copy of this portion. Okay. So, are you telling us that for the nested PCR, mas sensitive at mas specific? Bakit mas specific? Because of the primers, dalawang set yan. Bakit sensitive? Kasi dalawang rounds yung amplification na ginawa. Okay? So, ang kinaganda nitong traditional nested PCR is that, ang kinaganda ng nested PCR is that we remove or nawawala yung non-specific, non-significant amplifications na tinatawag. Itong part na ito, which is not significant. Kasi ang gusto mo lang ito. Tama? Ang gusto mo lang yung area na to. So, in order for you to harvest or amplify this gene sequence, this particular region, ladies and gentlemen, you make use of nested PCR. Makita mo na wala yung mga nasa gilid. Those are what we call the non-specific amplification. Okay? Ayun, that's for your nested. Wait, for the semi-asymmetrical PCR, you make use of your 2 primers. Okay? Dalawang primers ang gamit natin dito sa isang tube. Anong pinagkaiba? Paano mag-a-act yung first? Paano mag-a-act yung second? Yung first primer natin, ladies and gentlemen, can anneal at a different temperature from the temperature that we can use on the second primer. Again, when you say first primer, yung tinatawag mong outer primer. And when you say second primer, that is your inner primer. Okay? Okay. So, hindi na magta-transfer for the semi-nested asymmetrical PCR. Yes po. So, na-eliminate yung contamination for this type of nested PCR. Yes. Semi-nested, no? Kasi wala nang transfer na magaganap. Okay? So, that's it for your nested PCR. Thank you, Nest. Thank you, Nest. Okay, now we have the multiplex PCR or the multiplex polymerase chain reaction. So I want you guys to remember this. Highlight, bilugan sa longguhitan hanggang sa mabutas-butas ng papel. Sabi natin, multiplex, they make use of multiple sets of primers. Anong pinagkaiba nito sa nested PCR? Dito, yung nested natin gumagamit na ilang sets ng primers? 2. Pero remember, your master mix template mixture is subjected to 2 rounds of amplification. 2 beses natin nilalagay sa thermal cycler natin. For your multiplex PCR, ladies and gentlemen, 1 beses mo lang ginagawa. Yon, 1 beses mo lang siya ina-amplify, 1 beses mo lang siya ilalagay sa thermal cycler natin. Okay, maraming primers. Kung maraming primers, kung iba-iba yung sets ng primers na present, anong ibig sabihin nun? Ibig sabihin, you want to detect different gene targets. You wanted to detect multiple gene targets in your template or in your sample. Possible ba yun, Sir Joshua? Yes. Kunyari, ang isang organism, meron siyang 5 gene targets. Gusto mo ma-detect lahat ng gene targets na yun dun sa organism. Di gumamit ka ng multiplex PCR. Pero take note, hindi siya ganon kadali. Ha? Hindi ganun kadali, Sir Joshua? Syempre. Ang hirap kayo mag-design ng multiplex PCR reagent. Kasi dapat yung mga primers mo, since maraming primers ang gagamitin, kailangan number 1, yung primers natin, pare-pareho ng annealing temperature. Bakit? Eh, iba, di ba isang beses nga lang natin ilalagay sa PCR, isang beses lang natin ilalagay sa thermal cycler. Kunyari, ang primer set number 1 mo, di ba yung forward reverse, primer set number 1, ang annealing temperature is 45 degrees Celsius. May ganun ba? Example lang. Yung primer set number 2 natin, ang annealing temperature is 50 degrees Celsius. Primer set number 3, 65 degrees Celsius ang annealing. Okay lang ba yun for multiplex? Hindi. Kung 45 degrees Celsius yung isa, dapat 45 degrees lahat. Kung 50 yung isang set ng primer, dapat 50 degrees lahat. Hindi pwede iba-iba. Kasi nga, isang beses mo lang ilalagay sa PCR, yan. Ilang cycles? 35 cycles yan. Ay, totoo ba yun? Okay. So, medyo mahirap nga siguro talaga design ng multiplex PCR reagent. Number 1 consideration, dapat pare-pareho ang annealing temperature ng mga primers mo. Pangalawa, dapat, since marami na yung primers mo, may chances kasi na mag-anneal sila to one another. Anong mag-anneal to one another? Magdikit. Lalo na kung complementary sila. Kapag matchy-matchy sila, diba? Dapat hindi. Dapat hindi sila complementary. Okay? Okay, number 1, dapat pare-pareho ang annealing temperature ng primers. Number 2, dapat hindi sila complementary. Dapat yung primers hindi dapat complementary to one another. Kasi kapag complementary sila to one another, nako, kapag tumaas yung temperature sa annealing temperature, nako, pagdidikit sila. Kapag sila-sila na lang yung nagdikit, wala na mangyayari dun sa gene target mo. Wala na mangyayari dun sa template mo. Tama. Pangatlong consideration, real-time PCR machine po ba gagamitin or conventional PCR machine? Kung real-time PCR machine ang gagamitin, wala tayong problema kasi siya na bahala mag-analyze nung run na yon. Pero kung conventional PCR machine ang gamit natin, magkakaproblema tayo ng very, very light. Why? Kasi sabi natin, usually yung ginagamit natin for the analysis of the PCR product that has been thermally cycled by conventional PCR machine. Di ba, usually gumagamit tayo ng agarose gel electrophoresis. And ang principle nito, yung size ng molecule, size ng DNA molecules or ng nucleic acid molecules natin. Kung pare-pareho pala yung size ng amplicons. Again, when you say amplicons, these are the amplified fragments of DNA. Kung pare-pareho sila ng sizes, para saan pa? Hindi mo rin pala na-distinguish kung sino yung ganitong gene, sino yung ganitong gene, sino yung ganyang gene doon sa gel mo. Okay, so in developing a multiplex PCR reagent, dapat kino-consider mo number 1, yung annealing temperature, dapat pare-pareho sila. Number 2, dapat hindi complementary to one another yung mga primers, dapat hindi sila magdikit-dikit, dapat hindi sila matchy-matchy. And pangatlo, dapat iba-iba yung sizes ng amplicons para ma-check natin, ma-distinguish natin, ma-visualize natin ito via gel electrophoresis. Okay? Tandaan mo yan. Huwag kakalimutan. Okay? Ayun. Ay, Sir Joshua, question. A while ago, you told us na ang primers dictate the specificity of the polymerase chain reaction. True or false? True! So, are you telling us that multiplex polymerase chain reaction is sensitive? Yes, it is really sensitive like you have no idea. Okay? Ayan, dito tayo. So I have here a photo which I think, nakuha ito mula sa reference textbook natin. So as you can see, I have here your strain A, strain B, strain C, strain D. Okay, these are your primers. Okay, these are the primers for strain A, primers for strain B, primers for strain C, primers for strain D. Uy, lahat sila nakalagay sa ating master mix. Like, oh my gulay. So kapag present, yung mga genes na ito, doon sa iyong sample, what will happen, Sir Joshua? E di mag-aanil sila itong mga primers na ito doon sa gene na nandun sa sample mo. Kapag nag-anil sila, syempre ma-amplify yan. Diba? So, ang tendency, ma-detect natin lahat ng genes na ito. Okay? One very good example, ladies and gentlemen, of an organism that is being subjected to multiplex PCR right now is your SARS-CoV-2. SARS-CoV-2 is the causative agent of COVID-19. Ha, Sir Joshua, niloloko mo ba kami? Sabi mo kanina, ang SARS-CoV-2 testing, ang COVID-19 testing is reverse transcriptase PCR. Totoo naman po yun. But that, ladies and gentlemen, may also employ multiplex PCR. Pwede yun? Yes. Tandaan mo, SARS-CoV-2 RNA virus, automatic reverse transcriptase PCR yan. Kasi remember, RNA, if you want to amplify millions or billions of copies of your RNA, then use reverse transcriptase PCR. And Sir Joshua, if you want to raise the specificity of the RT-PCR, of the reverse transcriptase PCR, then we can make use of your multiplex. Meaning, maraming gene targets ang pwede nating i-amplify. Maraming gene sequence ang pwede nating i-amplify. Ay, totoo ba yun? Yes. Like, oh my gulay. Oh, kapag may kakilala kang medtech, promise, or molecular biologist, tanong mo, yung RT-PCR po ba yung reverse transcriptase PCR na ginagawa po sa sa SARS-CoV-2 ngayon? Pwede po bang multiplex yan? Yes, depende sa reagent. There are these reagent na isang gene target lang chinecheck. There are these reagents na 2, 3, 4. In my experience, ang favorite kong gamitin yung may 4 na gene targets. N gene, E gene, the RNase-P, and the RDRP. Uy, yun yung favorite kong reagent. But, you don't have to know the brand. Like, oh my gulay. Totoo, Sir Joshua. Yes, it actually helps determine kung anong strain ng coronavirus yung tumama. Oo. Oo, may ganon. Bahala kayo sa buhay ninyo. May ganon. Okay, so dito. Another example, your Escherichia coli. Your Escherichia coli. Naalala, di ba sabi natin, E. coli, your Escherichia coli, or your Escherichia coli. Remember, Escherichia coli, hindi lang po isang, hindi lang po isang basta Escherichia coli. There are different strains of your Escherichia coli that causes disease to humans. Oo, tanong niya sa molecular biology, itanong niyo sa bacteriology prof ninyo, Doc, Ma'am, Sir, totoo po ba marami pang strains ng Escherichia coli? Anong Escherichia coli? Escherichia coli lang yan. Dito. Bahala kayo na. Basta yung kasi yung tamang pronunciation. It's Escherichia coli. Ang arte naman, Sir Joshua, dito na nga. Merong enterotoxigenic E. coli, enteropathogenic E. coli, enteroinvasive E. coli, enterohemorrhagic E. coli, and enteroaggregative E. coli. Those are the strains of your E. coli that may cause disease or infection to humans. And pwede natin isubject yung mga yan dito sa PCR testing, sa multiplex PCR testing in order for you to detect the different genes that can be found in these organisms. Sir Joshua, question, is it possible na maraming klase ng pathogen ang i-check natin sa multiplex? Pwede. Okay. So, pwedeng isang organism, isang klase ng organism lang, pero iba't ibang gene targets? Check, multiplex yan. Sir Joshua, pwedeng iba't ibang gene sequences mula sa iba't ibang organisms? Pwede ba yun? Check na check, pwede yun. As long as maraming gene targets, as long as iba-ibang gene sequences ang gusto nating i-amplify, that is considered a multiplex PCR. Nakukuha ito, huwag kakalimutan, kailangan tandaan, hindi pwedeng tanggalin sa puso't isipan. Highlight bilugan sa guhit na dito tayo. Okay, and now we have the digital PCR, the last modification of PCR, you have your digital PCR. Ang galing-galing nito kasi ang dami niya applications, okay? Kaya niyang i-detect yung low levels ng sequences na matatagpuan sa pathogen mo. It can also quantify these low levels of pathogenic sequences. It can also express rare genetic sequences mula sa isang pirasong cell. And of course, it can perform clonal amplification of nucleic acids na gagamitin for the sequencing of mixed nucleic acid samples. Ano pang kinagaling nito? Kaya niyang i-quantify yung amount ng targeted nucleic acid na na-amplify natin during the PCR run. Totoo, yes, maski konti yung starting material natin, maski konti yung nucleic acid na na-harness natin mula sa DNA, kaya niyang i-quantify. Oy, totoo, Sir Joshua. Yes. Di ba you have your wells? Nadidetect niya kung sino dun meron. Tapos magpapakita siya ng graph. Kasi this digital PCR, kaya siya tinawag na digital kasi this PCR machine is connected to a software. This is not connected to a software. This is connected to a computer. And yung software na ginagamit for digital PCR is incorporated doon sa computer, so bahala na siya. Totoo, yes, I have here a photo that shows us, ladies and gentlemen, an example of how powerful the digital PCR is. Okay? So sabi natin, ladies and gentlemen, you have here your wells. Di ba dito natin lalagay yung sample natin? Tapos, kapag niron mo yan sa digital PCR or sa PCR machine that is capable of performing this modification, this digital PCR, siya na bahala mag-illustrate, magbigay na interpretation sa kada well na meron. Imagine, lalagay niya dito, positive to, well number 1, positive tong nasa B number 2, B number 3, posit din, posit. Ang galing, no? Tapos, pwede rin niyang i-quantify, bibigyan kanya ng values. Ang galing ng digital PCR. I'm a fan like, oh my gulay.